Good afternoon. We will discuss today about the measurement of chaotic flows using PIV. So, in this module we will discuss about different aspects of PIV, what is PIV, and then we will try to give a brief overview of this instrument, to be precise the I can say operational, the other working principle of the PIV. So, when we talk about the chaotic flows, in fact if we try to recall that in my last lecture we have discussed about the measurement of flow parameters in the turbulent flow environment. That means, I mean, if the flow parameters, flow properties and the to be precise flow velocity, which is not having uniform throughout this, uniform value, uniform magnitude in the field. So, I mean we have discussed that the flow velocity, which is having two different components in the turbulent flow environment, one is the average one, another is the fluctuating. Now, fluctuating part or fluctuating component. So, if we are interested in measuring the chaotic flows, then we have seen that either we can use measuring hot air anemometer. In that case we need to insert a probe and using 2 or 3 different wires at a time at a given point we can calculate the different components at that particular instant of time and at that particular point. We have also discussed about that the placing of a probe in a flow field leads to the local disturbances. Then the user or the experimentalist who is using this probe to measure the flow parameters to characterize the flow, he or she may not come up with the correct result because of this local disturbance. Now, to circumvent that problem associated with the measurement of flow characters using probes, another new technique came into the picture, and that technique relies on the optical method. That means we are using light source. We have seen from in last class that what we do, we use a light source and we try to illuminate a particular zone, particular portion of the flow field which is the zone of our interest, and from there we can calculate, we can predict the either quantitative as well or the qualitative estimate that which we are interested in. That means either we can calculate the qualitative, we can predict the qualitative picture of the flow field, or we can quantify, we can calculate the quantitative value of the flow parameter, and this qualitative as well as qualitative measurement is possible using this optical image based technique. In continuation of that, in this module we will try to discuss about the measurement technique using PIV. So, what is PIV? Before we go to discuss, before we go to briefly discuss about the I can say basic principle of PIV, we need to know what is PIV. So, this PIV is particle image velocimetry. So, this is PIV. From the name itself, at least we can understand that this technique, this method relies on the seeding of a few particles. That means we need to insert a few particles in the flow domain, flow field. Maybe it is boundary layer flow, or it may be the flow between flow in a confined domain. So, the particle image velocimetry, that is PIV, is nothing but the imaging of the particle instead of, so basically if we are looking for the velocity of the fluid, flow velocity, what we are doing, essentially we are trying to calculate the velocity of the particle which are seeded in the flow. So, this method, this technique relies on seeding the particles which are very important. So, the particle image velocimetry, that means we are using light source to illuminate a particular zone of the field, and we need to insert a few particles, and the particles should have a few important I can say characteristics, and that we should discuss those characteristic feature of the particle, and particle by measuring the displacement of the particle in the flow field, we will try to quantify the velocity of the flow. So, basically what is done, that this particle image velocimetry technique, that is PIV, this PIV I can say is a measurement technique for obtaining instantaneous I can say whole field velocities. So, even this method is very popular nowadays. This method will have a few disadvantageous feature that we will discuss, but what we can say now, that the method is used to measure the instantaneous whole field velocities, which is important for the turbulent flow. That means turbulence that is there, we need to measure the fluctuating part of the velocities. That means at a particular time in a given space, what are the different components, that is it is not a laminar flow, flow field is velocities or streamlines are not perfectly parallel, it is a chaotic movement, and we need to obtain the information about the flow field, whole field velocities. So, the PIV is a measurement technique for obtaining the instantaneous whole field velocities. If we try to recall that in the context of measurement of chaotic flows using LDA that we have discussed in the last class, LDA is another important method in which we are also getting qualitative as well as quantitative measurement of the flow field using light. Even for the LDA, that is laser Doppler anemometry measurement technique, we need to have a few seeding particles. So, similar to that what we have seen in the context of LDA, in this particular method as well, we are we are interested in measuring the instantaneous whole field velocities by seeding a few particles. So, the particles will be seeded in the flow flow field. So, this is the PIV technique. Now, this technique, it is based on well known equation, that is speed equal to distance by time. So, that means the particle will move from one location to other location in the flow field, because there is a continuous flow. If we can somehow track the particle in a given location, and if we know the time at that point, and if we try to capture the same particles in different other location, and if we know the time taken by the particle to traverse that particular distance, we can obtain the speed of the particle. As I said few minutes back, that PIV measurement is essentially measurement of the velocity of a particle, that is called particle image velocimetry. So, by knowing the velocity of the particle, we are predicting the velocity of the fluid. So, the speed is nothing but distance by time, and that is used again in this method to calculate, to predict the velocity of the fluid. Okay. So, now we will discuss a few important points. So, when we talk about particle image velocimetry or PIV in short, there are two different parts. First one is the setup and the working principle of the setup, and then finally another important module of this measurement technique is the imaging. That means, if we just go to the previous slide where we have written particle image velocimetry. So, that means we need to capture image of the particle, and from the analysis of the image we can correlate what will be the velocity of the particle. So, there are two important parts associated with this measurement technique. First part is the construction of the system and the functionalities of different parts associated with this system, and then second part is the image analysis, which is very important. So, today we will try to discuss about the basic principle focusing the function of different parts associated with this system, and then we will move to see the analysis of the images which are captured from the setup. So, the first part is the working principle. And I will write important points which are which you need to know in the to know the which you need to know about the PIV principle. Now, before we go to discuss about the important points, what we need to do, we need to know the schematic. So, I mean what we need to know the different parts associated with this system. So, now I will try to depict the schematic diagram of a particular PIV system. So, if we try to now write that we will have so this is, say we have flow from the top, so this is the flow direction. So, liquid or any particular fluid is flowing from the top, and this is the confined domain through which the fluid is flowing. Now, what we can do, we can illuminate a particular portion, say this is the portion we are trying to illuminate, and we have seeded particle like this. The number of particles will be seeded in a particular flow configuration, that will depends I mean on so many many factors. So, this is the confined domain. Liquid or any fluid is allowed to flow from the top to the bottom, and I mean the liquid is flowing or fluid is flowing, and we are focusing our attention on a particular zone in the flow field, and we can see that the fluid is now shown that fluid is now seeded with the particles. So, that can be seen. Now, importantly, if we can illuminate that particular portion. So, we are trying to illuminate this portion using laser. So, this is lens L, and light is falling on the lens using a laser source. From a laser source. So, this is laser source from which light is coming. The beam is taken through a lens to split into two different parts, so that we can project, we can focus our attention on a particular zone, and that is what is seen from the schematic. And what is done, the particles are, that is what I was telling, the object is, we have now illuminated a zone in the flow field using this laser source. What we need to do next, we need to capture the image of the particles, and then only we can correlate what will be the velocity of the fluid by calculating the velocity of the particle through different numerical techniques. So, what we need to do next, that means in this setup, the setup is not complete until we are now installing another important part, or we are now until we are integrating another important part with this system. That means we need to have one camera, and which will capture the image. So, one camera will be there, and that will be captured. This is the camera. And this is the light sheet. Right, and the small particles are the seeded particles. So, these are seeding. So, from this schematic, at least it is clear that this system is very simple. System is a very simple system, in which we have one light source, laser, one lens, then one camera. So, when any particular fluid is flowing through a confined domain, now through a confined pathway, we need to illuminate the fluid, illuminate a particular zone within this flow field, and if we illuminate it, we can see that the fluid is now seeded with the particles are seeded in the flow. Now, the zone which you are illuminating using the light will be captured by a camera. So, this is what is the working basic principle. So, now I will try to write the basic principle. I will try to write, and that will help you to understand even in a later stage that the light will be used to illuminate a particular portion, particular zone of the flow field, wherein we have a seeding particle, and that particular zone that I have marked by the light sheet, that means a light sheet, a particular zone is illuminated, and that will be recorded by a camera, and the camera will be located right angle to the incoming light source, at a right angle to the incoming light source. So, basic principle is that first one is that seeding. So, particles are seeded to the flow, and this method is known as seeding. Seeding. Different types of seeding are used, and that is depending upon the nature of flow to be investigated. So, this is the basic preliminary thing. We need to add a few particles to the flow, and this process or method is known as seeding. Different types of seeding are used depending upon the type of or nature of flow to be investigated, depending upon whether the liquid is density, liquid viscosity, and so many other things. So, depending upon the flow to be investigated, different types of seeding are used. Fine. Next important point is that, although I will discuss in detail, but at least for this for the time being, the seeding particle are chosen to follow the flow and in order to, which is very important, and in order to detect their movement. Right. So, this is important. Next is a few important. So, this is today what we are trying to know only the basic principle, and then knowing the basic principle, in the next we will try to know the image processing. That is another important part with this of this technique. So, this is important. Next is what you can see, an area of the flow field is illuminated by a light sheet, and which is generated, which is generated by laser source. Laser and other optical component. So, this is, so an area of flow field is illuminated by a light sheet which is generated by laser and other optical components, that is what we have seen. So, we need a laser source, we need lens and different other accessories, those we could not show in the schematic, fine. So, this is important. Now, important is the light sheet is not permanent, but pulse to produce a stroboscopic effect. What is that, I will discuss, and freezing the movement of the seeding particles. The time between light pulses is the denominator of equation 1. So, if we now go back to the previous slide, this equation is 1, that means the time which is there in the denominator, that is nothing but the time between two laser pulse, light pulse, now two light pulses. So, the stroboscopic effect, that is very important, we have written that moving object can be represented by a, so I have written the word stroboscopic effect and also the freezing the movement of the seeding particles. So, that is why the light pulse is not permanent, but it is pulse to produced. So, that means the stroboscopic effect, that means the continuous the moving object is represented by a short series of samples. So, that means it is not continuous, we can the moving object can be represented by a series of short instantaneous movement. So, it is not a continuous movement. So, moving entire movement can be split into a number of short small movement, and that is nothing but the stroboscopic effect. So, this is we have understood. Next important is that now we have to detect, and for to detect the position of the illuminated particle, a CCD camera, that is known as charged coupled device, charge coupled device CCD camera is used, and the camera is placed, that is very important, camera is placed at right angles to the light sheet, to the incoming light or to the light sheet, right. So, we have one camera, that is what we have seen in the schematic, and the camera is placed at right angles to the light sheet, and this is CCD camera. Now, the camera is placed only to detect the position of the illuminated seeding particle. The next is the particles particle position will appear as light specks on a dark background. On each camera frame, right. So, the particle position will appear as a light specks on a dark background on each camera frame. So, next what we will do, the pulsating light sheet or the pulsing light sheet and camera are synchronized so that the particle at the instant of light pulse number 1 are registered on frame 1 of the camera and particle position from pulse number 2 are on frame 2. This is important to know. That means, the pulsing light sheet and camera are synchronized, so that it is pulsing and camera also. So, the camera so that the particles at the instant of light pulse number 1 are registered on camera frame number 1 and pulse number 2 camera frame number 2. This was not this was not possible in older generation of CCD, where we had to capture all the position on a given or a same frame but this is not the case with the present days CCD camera. So, now we have understood that since the particle positions is a pulse that is called stroboscopic effect, that is what we have discussed. So, we will have the continuous movement with a short number of instantaneous movement. So, we will have a pulsing. So, the pulsing light sheet and camera are synchronized in such a way that the particles at an instant of light pulse 1 will be captured or will be registered on camera frame number 1 on frame number 1 of the camera, and the particles position at pulse number 2, that means the next pulse will be registered in frame number 2 of the same camera. So, next is that the camera image we have captured. So, the camera image are divided into rectangular regions called interrogation areas. Or interrogation windows, or interrogation zones. So, that means the camera images are divided into rectangular region which is called interrogation areas or interrogation zones or the interrogation windows. And next is for each of these integration windows or zones, the first and the second pulse of the light sheet are correlated to produce an average displacement vector. Right. So, that means for each of these integration zones, the first and the second pulse of the light sheet are correlated to produce an average displacement vector, vector. And doing this for all integration zones produces a vector map of average particle displacement. Right. So, doing this for all integration zones produces a vector map of average particle displacement. Now, if we divide this average particle displacement by the time between two consecutive pulse, two consecutive light pulses, we will get the speed of the particle. So, next I am writing that dividing with the known time between the two images captured the displacement vectors are converted into a map so called map of so called raw velocity vectors. Right. So, what we have discussed today is that by knowing the displacement vector, and if we divided that displacement by the known time, we can dividing with the known time, the two images between the two images captured, the displacement vectors are converted. This is converted into a map of so called raw velocity vectors. So, what we have understood is that we have captured two images and we know the particles location when they have captured in a particular frame, and we know the and that is known as correlation. So, we are also capturing the particle position in the second frame, and we can correlate these particles by this we can correlate their average displacement, that is dividing. So, if we go to the previous slide where I have written that average particle displacement that we have, that we are doing. So, that means knowing the average displacement, if we divide with the known time, we will get the displacement vector, and that vector displacement vector will be converted into the map of so called raw velocity vector. So, now question is, there are important question that I would like to discuss, that now we know the average displacement and we know the time between the displacement, then from there we can calculate the speed using equation 1. Now, we will discuss the critical issues, that what we have understood till now, that next part is the image processing, and that is very important to correlate the velocity of the particle, and from there we will try to quantify the velocity of the fluid. So, the next issue is that images which are captured in different frames will be now correlated to obtain the speed of the particle, and knowing the speed of the particle we can calculate the velocity of the fluid, and of course instantaneous velocity at that particular point of time. Now, so this correlation and image processing is an important task with this measurement technique, and we need to know several important issues which are related with this correlation as well as the measurement of the fluid velocity, and those aspects we will be discussing in the next class. So, with this I stop my discussion today. Thank you.